Neo-Darwinists try to explain everything with the idea of adaptation, but today we know that's not the case. Neo-Darwinist literature is completely devoid of critical judgment. They give implausible answers, and the whole theory falls apart. An alarm is triggered if Darwinism is questioned. Organisms do evolve, but Darwin's theory is not consistent at all.
These are the words of the American philosopher Jerry Fodor, who very precisely criticizes the evolutionary theory postulated by Darwin and carried on by the Neo-Darwinists. As many of you know, we have discussed the theory of evolution several times and have highlighted its unclear and questionable aspects. Today we will approach the topic from a genetic point of view, specifically investigating some obscure points of our genome, and we will not be afraid to venture into new and surprising scenarios. DNA, the instruction manual that makes humans different from any other species on Earth, hides something mysterious, something that could reveal an origin different from everything we thought we knew. Well, before we start this fascinating journey, as always, we invite you to subscribe to the channel to support our work and to not miss our new videos. Mutations underlie all evolutionary processes. The DNA in our cells, in fact, is constantly mutating. This is a normal physiological process due to the error rate that occurs when enzymes copy DNA during replication. It is estimated that every day several thousand of our cells mutate spontaneously. Obviously, to these mutations we must add all those that occur when we are exposed to mutagenic agents, such as UV rays. However, the only mutations that can be passed on to offspring are those that occur in germ cells, in oocytes in women, and in sperm cells in men. Also, in this case, researchers have estimated a rate of about 60 mutations per cell generation. This is still a very slow process that, on one hand, is at the root of all the genetic diseases we know today, and on the other hand, is the driving force of evolution as we know it today. These continuous mutations, which are passed down from generation to generation, contribute to an inexorable deterioration of our genome. This is a very fascinating topic that we have already discussed, and for those who want to learn more, we covered this topic in a video that you can find on the channel. Before we go any further, let's make a promise. From now on, we will tackle very complex topics, but we will do it in the simplest way possible so that anyone can understand the mysteries hidden in our DNA. Following this promise, let's imagine our genome as if it were an encyclopedia made up of 23 different volumes. The volumes are our chromosomes. Of each volume, in reality, we actually have 2 copies, which are similar because they contain the instructions for building a human being. But at the same time, they are different because each pair of chromosomes is made up of 1 chromosome we inherit from our father and 1 we inherit from our mother. Every cell in our body potentially has access to all this vast amount of information, but in reality, they are only able to read a small part of it, depending on their specialization. A heart cell and a neuron, for example, only read certain books from our encyclopedia, specifically those related to their function. At this point, it's natural to wonder how cells are able to read the information present in DNA. The information, in order to be deciphered, is transcribed into RNA molecules, which are then translated into proteins, which are the molecules that actually function within the cells. This is where the first fascinating mystery arises. Our DNA is made up of segments of DNA, the genes. These can be coding genes, meaning they contain the instructions for protein synthesis, or non-coding genes, which do not have this property. Until some time ago, scientists believed that the only genes worth considering were the coding ones, that is, those able to transcribe the code into mRNA molecules, and therefore into proteins. But there was a very curious fact that they were not yet aware of. The coding DNA segments that make up the volumes of our encyclopedia represent only 2% of all DNA, and at this point, it was reasonable to wonder what the remaining 98% of our genome was for. Initially, it was believed that this did not have any specific importance, that it was just the result, in fact the proof, of all the byproducts of evolutionary processes. And so it was called junk DNA, precisely to indicate its insignificant nature. But this classification, however, was short-lived. To their surprise, researchers discovered that simple organisms, such as Habditis elegans, a small worm just a few millimeters long that lives in the mud, actually had a greater amount of coding DNA than a human being. How was it possible, then, that such a simple and basic organism had more coding genes than a complex being like a human? A human? Clearly, something was missing in this analysis. It would only be later that it would be understood that the role of junk DNA is much more important than initially thought. Even though some aspects still appear mysterious today and perhaps could be the key to explaining the mystery of humans. But let's set this concept aside for a moment. We'll come back to it later. Now that we have understood how DNA is structured, we can go on to analyze a first anomaly present in our evolutionary path. Currently, we have sequenced the genome of two of our distant cousins, specifically Neanderthal man and Denisovan man. Unfortunately, however, the quality of the genetic material is not sufficient to allow for an accurate comparison with that of humans. The only way to try to do a comparative study, therefore, is to analyze the DNA of our distant relatives who are still alive, chimpanzees. By comparing the two genomes, it was found that they differ by about 5%. So it would be just a small percentage that makes us so significantly different from monkeys. As we said at the beginning of the video, human DNA is made up of 23 pairs of chromosomes, 46 in total, while that of chimpanzees is made up of 24 pairs, 48 in total. Specifically, researchers have pointed out that in apes, the pair of chromosomes 2A and 2B fused in humans to form chromosome 2. This fusion is called Robertsonian translocation. In humans, we see this kind of event when chromosome 14 and 21 join together, as happens in individuals affected by Down syndrome. Because these individuals have two normal copies of chromosome 21 and an extra, although partial additional copy of the same chromosome 21 attached to chromosome 14. So they have one extra chromosome. So if this is a rare but still possible event, what makes this fusion between chromosomes, which happened millions of years ago and involved chromosomes joining, so mysterious? The answer is simple. Aneuploid individuals, meaning those who have one or more extra chromosomes, suffer from infertility. To be even more explicit, molecular biology does not allow the creation of a new individual from the union of two gametes, sperm and egg, that have a different number of chromosomes, because it considers this event an error that cannot be perpetuated. So, to recap and clarify the situation millions of years ago at an unspecified moment, a primate, in which the fusion between chromosome 2A and 2B had occurred into a single chromosome, incredibly met another individual of the same species, carrying the same aneuploidy, and by mating they gave rise to a new species. Why do we talk about a new species? Because as we said earlier, nature does not allow the union of two germ cells that have a union of chromosomes, because it recognizes this occurrence as an error. Therefore, the offspring born from two parents with a chromosome in their germ cells will no longer be able to mate and produce offspring with all those individuals who have an extra chromosome. This is why a hominid of the Homo sapiens species cannot produce offspring by mating with another primate. Although this experiment was attempted in the past, in particular the Russian scientist Ilya Ivanov tried to inseminate female chimpanzees with human semen, but without any results. What happened millions of years ago in a remote place in Africa is therefore undoubtedly a very rare event, and it is precisely the extremely low probability that this event happened and gave rise to our species that has raised many doubts among several researchers. To this, we must add another detail: the analysis of the chromosome in question, which has been subjected to precise examinations. It would have raised further doubts and uncertainties.
Monkeys have 48 chromosomes, but humans have 46, so evolutionists are left with a discrepancy in chromosome numbers. And they need a solution that somehow makes humans genetically more closely related to monkeys. They believe that two monkey chromosomes fused and formed a single chromosome, human chromosome 2. In fact, they found a sequence that seemed to support that idea. It's a stretch of DNA with some telomere sequences, like those found at the ends of chromosomes, in the middle of chromosome 2. They called it the fusion site. In a chromosomal fusion, there should be two genetic scars, and you should have a fossil centromere in addition to the fusion site. It turns out that the so-called fossil centromere is also located in the middle of a gene, a large protein coding gene. This supposed fossil would make it impossible to form a fusion, so the fusion is disproved.
In this rare and unlikely event, some researchers see evidence that humans and apes have no kind of common ancestor. Others, on the other hand, see in this a sophisticated genetic engineering intervention carried out by an external entity that somehow selected the human species, forever separating it from its ape ancestor. Certainly, if this were a natural event, we would be talking about something extremely rare and in some ways bizarre. Bizarre because all known conditions of aneuploidy in humans are rarely compatible with life, and when they are, they cause pathological conditions, as in the case of Down syndrome. We need to add another detail to this. We don't know in practical terms what the fusion of these two chromosomes entailed, or what traits this event gave to the new species, but it is highly unlikely, if not impossible, that this alone determined human peculiarities. The only thing we are certain of is that this forever separated it from all other ape-like beings, setting it apart as a new species. Now, whether all this is the result of the improbability of chance or of an external intervention remains a fundamental question to answer. What made man the unique being we know? Answering this question is certainly not easy, but by analyzing our DNA, a very interesting hypothesis emerges. The answer could actually come from an event outside our planet. But let's go in order and go back to the concept we talked about in the first part of the video, the one referring to coding and non-coding DNA. As we said, most of our DNA is non-coding, and for a long time it was considered useless. But right in what was defined as junk DNA, particular genes called jumping genes have been identified, which show some very unique characteristics. These DNA sequences actually have the ability to duplicate and copy themselves into different parts of the genome through a mechanism called retrotranscription. In a nutshell, it's like having crazy sequences inside the DNA that from time to time decide to create a copy of themselves in the most random parts of the genome, modifying it. These retrotransposable elements are believed to be responsible for major rearrangements that our genome has undergone, as well as the expansion of non-coding DNA. To simplify, we can say that these elements have the incredible ability to shape our DNA, bringing out new genes from unused DNA strands. However, these incredible elements, after taking part in the creation of the human blueprint, became deactivated. They built up mutations that shut them off. But there are some methods that can reactivate these little engineers and awaken them from their genetic slumber. Currently, the only place where these can wake up is in neurons. And when this happens, the individual develops neurodegenerative or neuropsychiatric disorders. But studies done on rodents have shown that the reactivation of these retrotransposable elements can also bring benefits or specific features, such as the formation of long-term memory. The stimuli capable of reactivating them seem to be environmental stress, high temperatures, and ionizing radiation. To clarify the concept, let's use the example of an encyclopedia. And let's pretend that our coding DNA is all the text present in the volumes that make up our encyclopedia, and that our non-coding DNA is made up of billions of apparently meaningless words. These particular genes have the ability to take individual words and insert them into the sentences found in the volumes of our encyclopedia, so that these sentences take on new meanings. If we add that the behavior of these genes can be influenced by external events, it undoubtedly opens the door to fascinating speculations and theories. It is therefore possible that our evolution has somehow been guided by events of which we are currently unaware. 2 million years ago, when a supposed diversification among hominids occurred, a star exploded relatively close to our planet and flooded the Earth with radiation. It is therefore possible that this radiation influenced our evolution. And if somehow this event spurred human history, triggering those changes in our DNA that created the peculiarities that distinguish our species, why didn't it influence all the other living beings present on Earth? The leaps between species, the mystery of the Cambrian period, and the sudden flourishing of countless forms of life, the evolutionary anomalies we find in our research, all of this could have been triggered by an external event that set everything in motion. These are all fascinating questions, especially since today we have discovered another aspect that has revolutionized our understanding of DNA. As we were saying, the coding DNA for transcribing information works like this. Genes encode DNA into RNA, and this is then turned into proteins. Well, it was thought that this was the only way to create information and that it was a necessary step. So much so that it was called the central dogma of molecular biology. In reality, we've found that things aren't like that. The reverse process can also happen, meaning that strands of RNA can in turn become DNA. This axiom is very important because it's one of the foundations on which the entire theory of evolution is based. A random event that occurs during the transcription of genetic material gives rise to a mutation, which then determines a trait that can favor evolution. But if we have reverse transcription events, that is, RNA molecules turning into DNA, it means that not all genetic changes happen through random mutations, but that these can be generated by the genome itself. The genome is therefore able to modify itself and give rise to new traits. If we accept this, however, all the assumptions on which the theory of evolution is based fall apart, and we should then ask ourselves what DNA really is. The answer could be that it is a code, the creative matrix of all forms of life. A huge encyclopedia in which all possible forms of life are written. A code subject to random mutations, but that at the same time can act upon itself. The impossible improbability of all those events that mark the evolution of life. They wouldn't become any less improbable if we admitted, even for a moment, that the building blocks of life followed a predetermined path. And if we accepted all this, then what is this external guide? Is it information traveling through the universe in the form of energy? Information that originated with the very origin of the universe? At the moment, we've only scratched the surface of a mystery that surrounds not only the origin of man, but life itself. Within us, the answer to all our questions might be hidden. We might finally explain the enigma not only of our origin, but of life itself. An enigma that seems more and more each day to connect us to something alien, something that permeates the entire universe. But that's another story that we will address soon. For now, let's stop here. We are really curious to know your opinion. Let us know in the comments what you think about it. And if you like the video, don't forget to subscribe to the channel because your support is essential for us. See you in the next story.